The following is a selected video from masterthecontent.com, where you will find an extensive video library of lectures for a variety of standardized admission tests. We offer over 600 hours of detailed video lectures for a multitude of standardized tests. Use our interactive in-lecture table of contents to find specific topics of interest. Work through numerous in-lecture examples to help you internalize concepts. To learn more, visit masterthecontent.com. Your career, our passion.
Okay, so moving on, what I wanted to point out on this slide right here is a few more things just to summarize what we've talked about as far as the structure of a nucleotide is concerned. Now, a nucleoside will comprise a nitrogenous base and a 5-carbon pentose sugar. Now, we've established that. That should be something that you have committed to memory at this point in time. Now, if we add a phosphate group to a nucleoside, we turn it into a nucleotide. So that's a key difference. Now, the addition of a phosphate group to a nucleoside turns it into a nucleotide. We've established that. Now, nucleotides will then move on and they'll serve as the building blocks for nucleic acids. So nucleotides are the monomers that are strung together in a polynucleotide chain, which results in the creation of DNA or RNA, which are very, very key biological molecules. So as you can see, we have the structure of a nucleotide on the screen right there, which consists of those major components we just talked about, the nitrogenous base, the sugar, as well as the phosphate group. Okay, so what happens in this case, as far as polymerization is concerned, like I mentioned in the previous slide, in one nucleotide, the phosphate group will be bound to the 5' carbon, as is the case in the diagram we have on the screen right now. This is the 5' carbon right here. And in a neighboring or an adjacent nucleotide, once the covalent bond or the phosphodiester link is formed, this phosphate group will actually attach to the 3' carbon of the adjacent nucleotide. So that's how polymerization occurs. You have basically repeating what we call a sugar-phosphate backbone that consists of repeating sugar and phosphate groups, and that is pretty much how the attachment occurs. Now, the most fundamental role of a nucleotide in DNA is obviously the storage and the encoding of genetic instructions. We know that DNA is responsible for encoding genetic information, which is then transmitted from one generation to the next. It would not be able to do that or perform its function were it not for these basic elements of structure that are referred to as nucleotides. Okay, nucleotides can actually also act as short-term carriers of chemical energy. Remember, we've only talked about the addition of one phosphate group to a nucleoside to create a nucleotide. You can add additional phosphate groups up to the creation of even nucleoside triphosphate, which actually can function or can act as a source of chemical energy. So that's pretty much what the synopsis, that's pretty much a synopsis of everything we've talked about thus far. As far as nucleotide structure is concerned, what we're going to do next is we'll move on and do a very brief example just to test our knowledge of what we've learned thus far in the lecture.